which is also Anupama, I think, flashed it yesterday. And then we're also going to go, if you know how to basically construct or predict these flows, then the next level would be, can we actually control them? Okay, so that's what you are going to do. So this work is a long-standing collaboration with the group of Kees Beher at the University of Dundee. Kees is a developmental biologist that did all the experiments. Guillermo was a student with Kees and now is a postdoc in Cambridge with Ben Steventon. They also did other experiments. And Alex is a PhD student in the group. Okay, so what we're going to do today is, so this is our system. Okay, so we discussed yesterday. So for us today, the living system or the flow is just a layer of cells that is the chick embryo at the beginning of development. You can see it from opening an egg. And it's very important to understand this stage of development because two reasons. The first one is that the chick system resembles human gastrulation, which is this very early stage of development. It's called gastrulation, and of course you cannot study this process in humans because the cells are inside the body of the mom, so it's really inaccessible. So biologists, what they do, they consider model systems or animal systems that are the closest, that they share basically the similarities with the human similar developmental stage. Okay, and the chick is one of the vertebrates that shares this process the most closer to human. So basically this is how you want to try to understand what happens in human. Why this is important, again, because this is when you establish your main body axis and bilateral symmetry, and this is also when the first germ layers are starting to form. So basically if you mess up this stage of development, it's very you're going to have very likely irreversible consequences. Okay, that's basically the biological relevance of looking at this system. And the physical relevance is that really this system is basically two-dimensional. You can really image. You put a microscope on top, and therefore it's also interesting to understand how basically can we learn new mechanisms of living systems looking at this basically in vivo tissue. Okay, so what we did, and if you really want to have it just a little bit of a broader overview, we are looking at this, first 15 or 12 hours of development, when you start from a disk of cells, two-dimensional, to now an elongated, symmetry broken, if you will make, at least geometrical symmetry broken system, and then if you wait 18 days, basically you're gonna get the full embryo, the full animal, basically. Okay, so. Well, what I'm going to do, basically, I'm going to first go through what I showed quickly yesterday. So how can we quantify these multicellular flows using coherent structures? And then we're going to understand how to predict these flows and eventually how to control them, okay? And, as usual, stop me anytime. I mean, I don't have, usually I try always to calibrate slides in a way that there is plenty of time for questions, okay? Okay, so general question is, we have this tissue flow. This is now very clear, so I'm going to go relatively fast. If you start looking at this basically velocity field, regardless of what it is that is moving, you can either have an Eulerian description of the flow, which is basically, for instance, you can look at, velocity fields and streamlines. If you look at velocity field and streamlines, you're going to get some sort of, plots like this, okay? However, the problem with looking at these plots, especially in this context, but in general, is that the geometry of these plots are basically frame dependent. That means basically, for instance, that think about this embryo is actually it's happening. At some point, the embryo starts drifting to the right. Okay, just because maybe one edge here detached from the substrate on which is originally attached, and then everything drifts to the right. So just that thing that can very likely happen is going to change completely the geometry of this flow. And in fact, you can see here that everything is sort of translating to the right. Okay, so now another embryo doesn't do that. Another embryo may be translating north. There could be relative rotation with respect to each other. So if you want to assess the material response of this system, you don't want to use these techniques because they depend on the frame. Instead, you think about it, the embryo develops in a way that really doesn't care about what kind of coordinate system am I picking to describe the motion of its cell. That's something that shouldn't really affect any conclusion on where things converge, diverge, or stretch, okay? And this is basically the notion of frame invariance, which is that, you know, the material response of a deforming continuum, could be a living continuum, but also could be a passive continuum, should be independent of the observer, of the reference frame. So the velocity fields, also the vorticity, the Q criterion, or the helicity, and all this kind of stuff are all frame dependent. So we want to look for quantities that are frame invariant to describe the material response of a flow. So one option to do that is basically look at Lagrangian trajectories. So you start saying, okay, now I want to, instead of looking at fixed and position, I'm going to go to the Lagrangian coordinates. I will remember if I start here, where I will go after certain time t. Now this must be very clear, and then if you take now the gradient of this flow map or trajectory map, then you can also understand how an initial spherical neighborhood is going to deform. This is what we did in the last few days. Okay, if you want to visualize this tensor a little bit intuitively or naively, you can just think of looking at this embryo and deforming a Lagrangian grid on top of it. So now the beautiful thing is that every square is labeling a patch of tissue, so you can follow where it goes, you can follow how it deforms, and that's basically the same patch of tissue. You just label every patch of tissue, right? Okay, so we've seen basically the deformation gradient is what popped up yesterday. And, well, I mean, I didn't tell you yesterday, but, yeah?
So these Lagrangian agents are cells or tissues?
They could be any, right? So it depends. In this case, the dynamics is given via velocity field. That means so you basically do images. They do images. They stay in cell membranes. So you have some intensity field of cell membranes, and then with this snapshot of images, you do PIV, which you reconstruct some tissue scale velocity. So it's pretty high resolution, but it's not cell scale resolution. Okay.
Higher than cell scale.
Higher than cell resolution, but we also have trajectories that cell resolution doesn't really change. It's really the resolution of the data is going to tell you how precise you can be in, attractors, repellers, stuff.
But it gives the track only if there are cells, because it is that the tracer, the fluorescent materials are attached to the membrane only, right? So not around the cell. So around the cell there could be extracellular matrix, and there may be dynamics around that also. But if just
this is an epithelium, so okay, this is an epithelium. But yeah, if you have a mesenchymal situation, then you have to decide what to track. If you want to track the nuclei or.
In this case, it is fine.
Yeah. Yeah.
So yeah, fine. So in that sense, like we have the information of the.
Yeah. You have information of the flow and of the cells, basically. Yeah, they move together with their membrane.
Yes.
Yeah. Okay, so an exercise that you can actually do is that basically, the largest eigenvalue of Cauchy-Green, the square root of that is the largest singular value of df. It's an exercise that you can ask always as an exercise. If you have a linear relationship which is like that, and A is a matrix, 2 by 2 in this case, but in general, and you ask maximize, what is the maximization with respect to x of norm of AX divided by x? Do you know what's the answer? So you take a matrix, multiply by a vector, get another vector, and I'm asking, okay, you can, the matrix is given, and you can pick the vector. What's going to be the vector that maximizes the norm of AX, which is the output vector divided by the initial vector? It's not the eigenvalue. It's the eigenvalue only if A has some particular property, symmetric, etc. But if A is a general matrix, like basically df, the answer is actually the largest singular value of A. Okay, so the largest singular value of A, so if you want to remember zeta t is equal df zeta 0, okay, so if you want to maximize df zeta 0 divided by zeta 0, this is the largest singular values of df, which is also equal to the square root of the largest eigenvalue of C. Okay, it's just anything. So basically, basically you can compute the Lyapunov exponent just computing df. You don't need to do even the Cauchy-Green. Okay, doesn't matter, it's the same. Actually check that it's the same and also check it mathematically. Okay, so basically that's what we did yesterday. So, okay, looking at this matrix is a little bit laborious and not really visually appealing. So I'm gonna look at the largest eigenvalue. And then we define basically the repellers as basically ridges of this largest eigenvalue. You can basically do the logarithm and rescale by time, but these are just, they will not change the topology of the function because the logarithm is a monotonic function. So the maxima of the f will stay the maximum also of the FTLE, or of lambda 2 will also say the maxima of the FTLE. That's basically what we did. Then we computed it for this system. We basically let it go for increasing time interval, and then you see that this system is going to develop basically two repellers, right? Which we discussed yesterday. I'm going to discuss them briefly in a second after I introduce the attractor. This is also what we did yesterday. So you can compute where things initially far away will converge to in a particular time interval, t0, t, and you can also compute the direction of attraction, which is where things, in which direction things will actually converge. And in this system we had one attractor. Okay, so basically what we did in this compression is using these Lagrangian coherent structures, which in the context of development, using basically just attractors and repellers, not elliptic coherent structure, parabolic one, computed specifically with the FTLE, we call this dynamic morphoskeleton. Because with these attractors, repellers, and domain of attraction, which I also introduced yesterday, you can basically sort of compress these spatiotemporal flows of, tissues, okay, or embryos. So basically what we did, we started from raw velocities, right, and we just compressed this dynamical system, if you will, into 3 units. So the repeller number 1 splits all these cells that will not be forming the embryo. The attractor marks the primitive streak, where things will go converge and then will enter into the third dimension to start to let three-dimensionality start to form, which is how you form your internal organs and our body. And then the repeller 2, we saw that basically was splitting the anterior part and the posterior part of the streak, i.e. the attractor, by splitting its own domain of attraction. That's basically what we did yesterday. Is this clear? Okay, so now it's beautiful because we have 3 units. So you have basically spatiotemporal system given over 12 hours, and we compressed it into 3 units. Think about it. This is basically what you do when you start looking at, classical dynamical system. What you do, start looking at fixed points, compute stable and unstable manifold, then you could look at limit cycles, etc. So you throw away all the rest and you retain all these robust ingredients, okay? This is basically what we are retaining. Yeah?
So the time scale, like, for example, repeller 1, so maybe that might be appearing earlier. And do they stay for like throughout the process, or do they disappear towards the end, or how does it work? Like?
They stay. So we can see that here.
Oh.
Right? Sorry.
So if you have slides later, we can...
No. We can see basically when, you see here, you see this guy forming and stays. It gets sharper and sharper, actually.
The second one.
Yeah. I mean, this is a noisy data set. It's really cells. Yeah. No, but let me actually show you. This is a very good point. So now that what I think, because we have a little bit of time, so I don't really have to go fast with these slides. But think about it: if you would start comparing these systems, and you wouldn't have some techniques like that, what would you be comparing? There is infinitely many things you can start comparing. You say, okay, what should I look? Should I look at the integrated velocity over the domain, over time, or the vorticity, or I don't know, some statistics of it? modulo the fact that they're all frame dependent. So that means basically that if just an embryo drifts because one edge is detaching, maybe this is due to the mounting artifact of thing, this is going to affect your metric. Okay, instead, all these guys are frame independent. It's okay, this by construction, they don't depend on which frame you use. And they are basically compressing this information into very robust units. This is, think about it, what would you do if you would have to answer, how do I compare these different systems? Okay, so just one thing, so these objects are the most robust features that you can extract from a dynamical system or a velocity field. And in fact, what you can do with very low resolution microscopy, so basically Guillermo Serrano in Cambridge in the group of Ben Stevenson developed basically a platform in which he can image 9 or 20 embryos at a time. That means compared to the data set that I showed you here is one embryo at a time, but you can have a much higher resolution, a resolution about a couple of cells size per pixel. So it's a very high resolution. So but you have to be very slow in scanning the embryo, and you can only scan one embryo at a time. So Guillermo instead said, okay, you know what, I'm just gonna give up on resolution, but I want a larger N. So he basically now can image 9, 10, 20 embryos at a time. Okay, and therefore the structures will be less resolved than in the previous case. But the interesting thing is that 9 different embryos, the shape of these repellers may change, but there are going to be always 2 repellers. Okay, sometimes there is a third one appearing. We are going to understand why is that the case, because after basically this body recirculates here, things will start separating, similar to the double gyro stuff that you have. So this is not really an artifact. But the long story short is that these are very robust. So now imagine comparing, I don't know, 9 velocity field over time. Okay, these guys will always be there. Okay, so this is a very strong features that you want to use to compare, noisy data sets. Okay, so what do we have now? We have basically 2 repellers and 1 attractor for this developmental process, and of course the same for the attractor. I just didn't show the attractor, okay, for robustness. Okay, so now we want to understand. Let's try to understand what are the mechanisms that generate this flow. I mean, to address this question, you really have to interrogate the biologists because they know what's going on. Okay. Okay, so what we want to try to do now, we want to try to understand, first of all, can we build a model that reproduces this tissue flow? Okay, so that you just, simulate like the models that Anupam described us yesterday. So can we build one to simulate or to predict these tissue flows? Can we understand what are the origins of attractors and repellers? What are the forces that are generating those? Okay. Then we say, okay, well, how does actually the embryo changes its shape from a disc into a pear shape? And how does it do it? This means basically you require some distribution of forces that shape your boundary, if you will. And can we actually control these guys? Because if you really know how they form, then the brutal way to understand if you really know how they form, then you say, okay, can I tweak them? Okay, so in vivo. That's what we are gonna try to do. And stop me anytime. So when you basically look at this system, again, remember this guy is a little disk at the beginning, at the initial time. There are 2 cell types. So there is basically the embryonic cells. Then there is an extra embryonic ring. These are the cells that will eventually be separated by repeller number 1. So they will not form the embryo. And within the embryo, there is some cells in the posterior that are different. They are called mesoderm. And the mesoderm cells are the engine of these systems. Whenever you have cell moving, it's because mesoderms are driving it. The active forces are usually in the mesoderm. Okay, this is basically just a way to explain mesoderm. Of course, genetically they are very different. They are different cell types, so we can go also in that direction. But okay, we don't need it to go that direction for this talk. Okay. Okay, so this is at the initial time. At the final time, that's what happens to the embryo. These cells are moving outward because they are crawling on a substrate. The extraembryonic tissue expands. You see there is an expansion here. The embryonic basically transforms into a pear shape, and this mesoderm basically goes to the attractor and internalizes. And strictly speaking, also recruit more mesoderm. You lose these engine cells because initially you're on this plane, then you go to the attractor, you leave the plane, but you recruit more mesoderm via induction on the plane. Okay, that's what's happening, but not important. So remember, 3 cell types: extraembryonic, embryonic, mesoderm. The mesoderm is the engine. Okay, and this mesoderm starts from the posterior at the beginning and then goes to the center line. And there is actually also these embryonic cells. So I'm coloring in the light blue. This is a darker blue, light blue, and white. Okay. Okay. So now, what are basically the active processes that are driving this system? So there is basically active intercalation into the mesoderm. Intercalation just means basically that cells that are initially far, they converge. So therefore, they basically exchange neighbors, and therefore they converge, and then they do a kind of a saddle flow. This is what happens. These cells basically do this, and then these two that were initially in contact, they're not in contact anymore. That's basically, they do it by contracting and modulating tension in their junctions, if you want to understand. So that's what's happening in the mesoderm cells. There is a lot of intercalation. Then there is, in this system, there is a lot of apical constriction. So cells basically will decrease their area. And then eventually here there is going to be an ingression, but in general they will decrease their area. So they will contract their area, and therefore the packing, so basically you're going to have a higher cell number density over time. Yeah.
So that disappearance of the cell is going to start developing.
Yeah.
That's what it is.
Yeah, that's, but in general, there is also reduction of apical area of these cells. So the cells basically from a little cylinder, they will squeeze, they will get longer basically. So then there is the active ingression. So basically at the streak, a lot of cells are actually ingressing. So you see that there is a flow of cell, and then these cells are basically, they keep flowing from the sides, and then here they are going to disappear in the third dimension. That's another process that is driving this system. And then there is these edge cells that are basically just crawling on a substrate. This system is very fascinating because all this piece of tissue is suspended. So there is, this is the extraembryonic blurb. So you have this segment of tissue, is that there is no substrate here. Then there is all the epithelium or the embryonic tissue here. And then of course it's going to be symmetric on the left. And then this little peripheral edge is attached to a substrate, and here cells basically using myosin, they develop protrusion and they pull these cells outside. Okay, that's basically what's happening. And wherever you see this motility or wherever you see this activity is driven by myosin. So myosin is a molecular motor that basically synergizes with actin, binds with actin, and contracts. It's basically what we do in our muscles, basically. Yeah.
So the measurement has been made on myosin level, or is there some other protein like Wnt or FGF is also?
You don't really need it at this level.
I'm talking about in experiments when they did observations. Like, of course, from tracks you can see, and also indirectly. But did they ever comment?
There are other studies on Wnt measurements and FGF, but those are more for differentiation in this case, because really myosin drives the system. This was established already in maybe 10 years ago. Yeah. So basically now, so this is what's happening. Now I'm telling you that wherever you see myosin, there is higher activity. So myosin is required to do this active intercalation because you need myosin to contract these junctions. You need myosin to contract your apical area. Just contracting all these junctions together, or these internal filaments, will actually shrink this apical area. So, but to do that, you need myosin. To ingress also, you need myosin because actually you have to shrink it so much that eventually you reduce it to a point and ingress. And you need myosin at this edge to move, okay? And if you basically do the experiments, this is how myosin looks like. So basically, this is at this stage, the embryo, wherever you see cyan, this is the active myosin. So you see there is myosin in the embryo, there is more myosin at the attractor, there is no myosin in this extraembryonic region. This is actin, so it's another basically marker that basically, so myosin and actin will bind together, and if you have active myosin, these binded filaments will contract. Here basically you have only active, no myosin, so this tissue is a passive region. Here instead you have both. Those are basically just superimposed. So whenever you see both, that means that there is both actin and myosin. I mean, it's not good quality, but you would see also the red here. Here there is only actin, non myosin, and then there is this myosin on the ring where cells are actually migrating outward. Okay? This is the only biological slide I have. Now we're gonna, I mean, maybe there is a little bit of quantification in the next slide. Questions? Okay. So what we want to do, we want to try to build an active stress. Okay, so in this tissue there are basically three regions that I already told you. So if you zoom here, there is no myosin. Okay, so this is more of, we think of this as a passive region. Okay, there is no myosin. If you go in the embryo, there is a little bit of myosin. You see that there is cyan, and the myosin is pretty much isotropic. It's not much oriented, because myosin is really like in active nematics. You have alpha, which is the parameter of activity, a strength, which now here is not going to be a scalar, a constant, it's going to be a scalar field, because some regions will have more contraction or contractility than other regions. Okay, that's basically the scalar field, the myosin scalar field. And then you want to understand nematic order parameter and orientation. If myosin is distributed more on a particular direction, which means basically that, I don't know, if I have cells, say if I have a cell and myosin is more activated in this direction, so there is going to be myosin in this region, but then you also want to say, hey, look, I mean, there is going to be a preferential direction of myosin in this direction, therefore the contraction is going to happen in this direction. So that's exactly how people model active nematics. Okay, and this is these bars that you see here. The yellow bar, we're actually marking from experiments the alignment or anisotropy or directionality, if you will, of this myosin signal. Okay, and you can see basically that at the attractor there is a lot of myosin. This is why the scalar field is brighter, but there is also a lot of orientation. So cables are aligned, or your myosin is distributed preferentially in the perpendicular direction of the attractor. Okay. So this is basically what you can measure, what they measured, and therefore let's, yeah.
So yellow arrows are just average quantity.
Yeah, they are average quantity. Yeah. So you do basically myosin imaging of this, and then you do Fourier transform, and you consider the mean, the anisotropy, and everything. Okay, so what do we need? We need a scalar field myosin activity. Okay. Then we need a nematic tensor. Are you familiar with the nematic tensor? Should be familiar with the nematic tensor. Basically, this is what's, Anupam described it quickly, but again, this is exactly what I said. So if you are, this is going to tell you the orientation of the nematic director, which in this case is the myosin orientation, and S is the nematic order parameter that tells you that goes to 0 when there is no oriented myosin, or it goes to 1 when all the little cables, basically, if you do an average, let's say if you do an average basically on a control volume, and these are all the cells, right? So then each of them will have a particular preferential orientation of myosin. You do this average, and you're gonna pick this is the average value of these cables. Just do the average, and then S is telling you, if S is equal to 0, that means basically that they are all randomly oriented, so there is not even a uniform, a particular, well-defined mean. But if myosin is equal, if S is equal to 1, instead that means that all these basically cables, or unit cables, if you can call them, or the orientation of myosin in each single cell is aligned with the mean. So the mean, all the cells are basically contracting myosin in the same direction. That's what it is.
It is very similar to the way N director field was defined, if you remember that again. So yellow symbols are the director field, right?
Yeah. Yeah.
And then from that you can construct the Q field that we have seen.
Yeah. I mean, are you familiar with the active nematics, liquid crystals? Okay. Yeah, that's basically the same tensor. Okay, so these are three fields, and therefore now we can just build the active stress. We're gonna say, okay, myosin is going to be a fraction of activated myosin, it's basically a concentration. Alpha is going to be a scalar that maps myosin to stress, okay? And then there is going to be an identity tensor that accounts for the isotropic contribution of forces or stresses. This could be associated with ingression. Remember, ingression, if you have a lot of myosin here, you're just going to contract isotropically, so you're going to generate an isotropic active stress. And instead, if you have the anisotropic one, that's what's going to actually model the active intercalation. So, okay, I'm going to actually contract here and go in the other direction. Okay.
So,
yeah.
So from the data, can you differentiate how much isotropic and how much anisotropic?
Yeah, that's exactly that, right? The mean is going to be the mean. You can basically, if you have a scalar field, you can understand what's the average of the scalar field, and then you can also ask, is there a preferential orientation and how much is distributed in that particular orientation?
Okay, I see. So you do a vector sum and scalar sum, and that will give you the
yeah. There is a PRE paper that now I don't remember the authors and the name, but it's in the citation in the paper, in our paper cited, in which you can extract these quantities from the measurements of myosin. Yeah. Okay, so are we on board with this? Okay.
So please stop if you do not move, because this is that it is going at a slow pace, so you can stop and not.
Is it clear why it's isotropic and isotropic and okay? It's an active stress. We already, yesterday we've seen many types of stresses, right? So this is just, a stress, right? So there is going to be an anisotropic part. An isotropic part, that means there is no preferential, all directions will experience the same value of this stress, and then this instead modulates this amount of stress in the particular direction based on, and also accounts for the fact that you have cables and how much are they aligned with each other, basically. Okay, so now this system is very this system basically is very viscous because it's basically made of cells that are basically sticking with each other. Reynolds number is very low, so you can just kill the inertia term, and it's just going to be a static force balance. So the divergence of the active stress, which is what I just defined, should be balanced by the divergence of the viscous stress. This is what you have also in Navier-Stokes, Laplacian of velocity. Here you have this additional term because the system needs to be compressible on the plane, because cells are basically going into the third dimension. So this is not an incompressible case, and also because the extraembryonic cells are stretching. Okay, so if you account for compressibility, there's going to be this other term that pops up in the passive stress. Okay, and this is just the divergence of the active stress that I just defined. Is it clear? Do you know how to compute the divergence of a stress, guys? Yeah. Okay. Okay, so now what do we need to close this system? To close this system, we need, this is basically two equations, because it's a force balance and V is two-dimensional. And therefore, we need an equation for M and an equation for Q, the nematic tensor. Okay? So now we have to build these two equations. So one is the equation for the myosin dynamic. So myosin is basically now the fraction of activated myosin. So think about it that, each cell, so let's say this is a cell. So this cell inside has myosin, but is not activated. There are all these protein, actin, myosin, so myosin is not activated. But then it can be recruited to where the cables are, or they could be basically on the junction or internal, etc. And if myosin gets recruited, then you start having active myosin, which generates stresses. Okay, so this M is just the fraction of activated myosin. It goes from 0 to 1. Okay, why I did that? Because this is easy to have, and we did that because it's easy to have a dynamic of M that is bounded from 0 to 1, and then I offload the transformation between M to stress to a constant alpha. Okay. So now if you have a cell that basically has a lot of active myosin and this cell moves, it's going to carry the myosin with it. So basically that means that there's going to be an advection term that carries myosin, okay? Then there is going to be basically an activation. So you say, okay, I can activate myosin from inactive to active, and I saturate at 1, because at m equal to 1, I cannot activate more. That's all I have available, okay? So there's going to be an activation term, and then there is going to be a detachment term. This is the detachment between actin and myosin. And people did experiments showing that basically if you have actin and myosin, they are in contact. Depending on how much you are pulling this actin and myosin, depending basically on the tension that is on this actin and myosin, the dissociation rate is tension dependent. The more tension you have, the less you dissociate. This is a so-called catch-bond mechanism. It's basically a mechanosensitivity process. Okay, so this basically tells you that the coefficient that governs the detachment, so the detachment rate, is going to be dependent on myosin and has this exponential form, consistent with these experiments. Okay, this m should be tension, but basically if you do a little bit of calculation, you can see that a long time scale, which is basically, you can find it in this paper, a long time scale, basically myosin is a good proxy of tension. So that's basically it simplifies the equation. Okay, so that's it. So basically this is the detachment, because remember, these cells are moving. They can recruit myosin myosin to become active from their cytoplasm, from the interior of the cell. Then you start stretching this cell because they're moving all together, they feel the effect of other cells, etc. And then when they start stretching, these actin and myosin here that are on these basically junctions, they say, okay, should I dissociate or not? And they dissociate depending on the tension that they feel. Okay, that's basically this term. And the last, and the last bit is that, so my actin and myosin, what they do basically, if you start contracting cables, what happens is that the cables in the cells near you will be under tension. If I contract, I will put my friends under tension, okay? And that basically generates, again, this is basically another derivation that you can see on the paper, but this generates basically an accumulation of myosin in the other cells. Pull, the cells will be more under tension, and with these catch-bond mechanisms, basically this generates further increase of myosin in the other cells, that you can actually show that this is equivalent to a diffusion of myosin, effective diffusion of myosin. It's a diffusion modulated by the Q tensor. So this thing can diffuse only if you have cables. That means that the nematic order parameter is different than 0, and then it can diffuse along that direction. It's not a regular diffusion. This myosin cannot diffuse isotropically. It can only diffuse on the cable. Okay, so this is something that you can, it's a directed diffusion along the nematic, cables, or along the direction of the cables.
Like if Q is there, then only.
Yeah, if Q is Q, it's basically the second derivative along the direction of the cable, along phi. So it's basically a directed Laplacian. It's not an isotropic Laplacian.
So it will be projection kind of thing.
Yeah,
yes,
yeah,
take Laplacian
yeah,
projected.
yeah,
Okay.
yeah. Okay, so this is basically the minimal model for this dynamic, and what we found basically, we found this thing. This is basically now a few years ago. We found basically that if you study the equilibria of this PD, again, what do? You have a PD in this case, not an ODE, look for equilibria. That's all you do, always, right? Because otherwise, what do? You just look at this equation. This is complicated. So if you look at the equilibria of these equations, are basically 3 fixed points. There is one fixed point that is at very low myosin, activated myosin, basically very low, that is stable. There is one that is also stable at very high myosin, and there is one that is unstable. Just look at this equation that we built in collaboration with the experimental collaborator that told us a lot about this, biology. And, interestingly, we thought, okay, this is exactly what's happening in the extraembryonic. There is no myosin, so it's kind of, it's basically almost with no myosin. This is a passive region that stays passive, unless you're going to overcome this sort of threshold myosin. And if you overcome this threshold myosin, you're going to go to the unstable equilibrium, and this is actually, sure enough, it's what's happening in the attractor. Okay? So basically, we thought that basically an instability of the active stress can actually be a driver of this morphogenetic process. So the embryos may be leveraging this instability of myosin to drive the flow. And in fact, we showed that in the embryo, this prediction was done with the model. We showed actually in the embryo that the myosin increases over time. So it's consistent with this instability. Is this a little bit clear? So there, depending on where you are, you can either go and stay passive, very little activity, right? But if you overcome a threshold, you're gonna go basically towards the higher value of myosin, which is actually what you observe in the attractor or the primitive streak. Okay. We need one more equation for Q. This is basically an equation that is very typical in active nematics, except this term. So Q is basically, again, Q is going to carry the information, two information for every point on your tissue, Eulerian coordinate. You are basically, at any point, there is going to be an average alignment, phi, and a sort of how much all the others are aligned with the mean. Okay, if they're all like this, S is equal to 1. If instead in another region all the things are just randomly aligned, phi is not defined. It's no problem because S is equal to 0, so here Q is equal to 0, which means that sigma active, the anisotropic part is equal to 0, because that's basically, remember, sigma active was alpha m times identities plus Q. So that means, okay, whenever I don't have aligned cables, I don't have an anisotropic stress. At best, I can just contract isotropically. Okay, so the equation for M, we did it. Is it in here? So what I just showed. And now you can have, the equation for the cables. I mean, similar to what we've seen before, is a patch of tissue moves, it's going to carry with it Q. So that there is the advection. Then there is going to be two terms that are basically the shear and rigid body rotation. So this is basically standard in active nematics. So if you basically have that, let's say that for instance here the cables are all aligned in one particular direction. If there is a vorticity in this system, of course all these cables will rotate. Okay, so vorticity, rigid body rotation, vorticity reorients, which is these terms. Okay, and then dd is the rate of strain tensor of the velocity. This S is the nematic order parameter, and this models basically this fact. So basically, if you have basically a shear flow, right, like that, this shear flow is either going to align cables in the direction of largest eigenvector of the shear, or destroy the order. If you start, for instance, from cables initially ordered horizontally, this is going to just flip the order of the cable. Okay, this is also very similar to many of the things that Anupam was telling us yesterday. Actually, you can show that if you remove this term, this guy, remember what are the information that are contained in Q, S and phi. You can actually, so I think Luca Giomi decomposed using Pauli matrices, decompose the equation for Q into an equation for S and to an equation for phi. And if you do that, the equation for phi is exactly the Jeffreys equation. So basically, this is exactly the Jeffreys equation for the angle modulated by the nematic order parameter, which is beautiful. It's important because this allows you to play also when the cables or the orientation is not defined, like the defects and stuff like that. Okay, so now the problem is that if you just use these three terms, this system wouldn't reproduce observation, because remember what happens at the streak is that the streak, you remember we observed experimentally that the cables at the later stage at the streak were perpendicular to the streak. But the flow is such that at the streak things are converging. So this is cables, which means basically Q. And the flow here is such that things are coming in and also going out. So this is basically precisely this saddle type scenario. But the problem is that this is an unstable configuration. Okay, so I'm going to flip the cable and I would just turn the cable parallel to this streak. Okay, so this self-induced flow, because this cable contracts, will generate the flow. This is the velocity field. Okay, and this thing would actually self-destroy order at the streak. Okay, so you need a mechanism to sustain order. Okay, and this is what we basically constructed. We said, okay, if there is going to be two terms. So basically, the active mechanism is this one here. It's going to say, if I'm in a region of the system in which I have a little bit of order, that means that S is greater than 0. S greater than 0 means that Q is non-zero. So if I have a little bit of order, and now I start contracting in this direction, so I have a lot of myosin there, so I start contracting in this direction, which is S. What happens, basically, I'm just going to create even more order because I'm going to redistribute myosin and redistribute myosin more in the direction of contraction. So this is basically going to increase the nematic order. Okay, so think about it. Basically, you put, maybe a little bit imprecise, but maybe intuitive way of thinking about it is that if you have a bunch of people with loose ends, if someone pulls, you straighten up all the ends. Okay, that's basically, you can think of this. It's not exactly like that, but that's basically the idea. It's called basically active alignment. So the active alignment is that you start contracting this junction, and then you redistribute myosin in the direction of contraction. So this is where the myosin is going to be more polarized, and therefore, because you're polarizing more in this direction, you're going to increase the nematic order parameter. Okay, so basically we call this active alignment. And then you just have a passive relaxation. So this guy would just, if in absence of all the other guys, this guy would just destroy order, because cells are basically dividing, ingressing, intercalating in a way that you just destroy order. Just is a system basically out of equilibrium. Okay.
What is this small s?
Nematic order parameter.
Small s is the negative.
Yeah.
Three-factor.
Yeah. This is just, if you think of this as an equation for S, this is just decreasing S. Yeah, it's a decay, and this is because if you really think of this system and you look, there are a lot of division, ingression, etc., and these are just things that are randomly happening, and therefore they destroy order. If you would have cables all in a particular direction and cells divide in these regions, you're going to destroy this thing, because you have to reform junctions and stuff like that. Okay.
So S is both space variant and time.
Yeah. Q space and time variant, V space and time variant, M space and time variant.
So usually for decay you use a linear term unless you need to maintain.
There is. Yeah, I mean we show in the supplementary that basically you can. This is not changing anything. You can pick any functional form that is reasonable. We picked this because it was standard in active nematics to have a quadratic form, and I think the reason was for maintaining S bounded in the active nematic field, not in the bio thing. I think there are a couple of papers from Amin or Luca. So we kept the standard term, but it's insensitive to that form.
Keep it bounded, otherwise linear term will not be taken. Because there is a linear test term that is S and DD. Maybe that is there is because
there is a linear term.
Growth, and then that's it. You need a higher order term.
Yeah, but also if you pick it linear, in this case, it doesn't really matter much. Yeah. Okay. Okay. So now we have these three equations. The only thing that we are missing is the initial condition and boundary conditions. Okay, so boundary condition is basically simple. For the velocity, we are just gonna keep from experiments this outward velocity at the edge. You can just pick the velocity from the experiments. Okay, the cells are just pulling at this outer edge, and they pick that as a boundary condition for the velocity. I pick no flux for M and phi and Q at this outer boundary, the least invasive boundary condition, because there is not really a known source or fixed value, etc. Okay, so then initial condition, the boundary conditions we are done. We need initial conditions for M and T, for M and Q. Okay, so now we get it from experiments. We know that at the beginning, you remember the disk that I showed you at the beginning? At the beginning, if you really measure myosin, there is more myosin in the posterior where the mesoderm is. That's basically what we found at my initial plot of this disk. So there is going to be higher myosin in the posterior, and if you do the quantification, you see that there are cables aligned in the posterior, So there is going to be higher nematic order parameter in the posterior, slightly higher nematic order parameter in the posterior. That's basically our initial condition for the cable and myosin. And we also know in general that there is more myosin in the embryonic region compared to the extraembryonic region. That's all we know, right? And so this is our model initial condition. So the embryo is circularly at the beginning. The boundary of our PDE is here. Here cells will move outward with a fixed velocity. There is more myosin in the embryonic region than in the extraembryonic. We know that. There is also instability. There is a little bit more myosin in the posterior here where the mesoderm is. It's not really good quality, the projection, but you can really show this experimentally. And there is higherly, a little bit of more aligned cable in this posterior region. That's what we basically have from experiments. So what we want to do now, we want to say, okay, from these initial conditions and this PDE, if I solve it for, I don't know, the next 15 hours, can I reproduce the fields that I observe in the embryo in the next 15 hours, okay? And that's what you get, basically. You get that if you solve this equation, this is going to generate a velocity, which I'm plotting here. So it's basically this embryo, so this model generates a velocity, it's going to generate the vortices, it's going to generate the embryo shape change from a circle to a pear. It's going to generate the extraembryonic that expands, and it's going to generate negative divergence at the streak, which is where cells ingress. Okay, and the interesting thing is that this boundary is not the boundary of our domain. The boundary of our domain is here, fixed. That's basically the boundary of the extraembryonic region. So this means that this model is reproducing a correct or a plausibly correct, basically, distribution of active forces that actually change the shape of the embryo from a circle into a pear. Okay, so this is an interesting observation. First, you can match all these data. You can look at experimental velocities. You get these vortices. You can get negative divergence at the streak, and all the other possible combinations are all in the reference. You can also actually check if this model is reproducing the right distribution of active stress. Okay, so this is saying, okay, now this is the evolution of myosin. Remember, you have myosin is the amount of active stress modulo the constant. So basically, myosin dictates how much active stress you have. So you start from higher myosin in the embryo, no myosin in the extraembryonic, more myosin in the posterior, and later on you see basically that myosin increases because there is an instability, and higher myosin is present, actually develops more at the streak due to the instability, which is exactly what we saw in the experiments. When I showed the myosin distribution, there was this very high cyan concentration at the streak. Now, this is what happens at the final time. The model knew all information at the initial time. Now, if you do the experiment at the final time and you try to measure active myosin, you get this, which is very similar, actually, to that. That's basically the streak. There is a lot of myosin here because these cells are crawling. We got rid of this complexity and we just said, okay, let's actually use just the resulting outward velocity. So otherwise you have to put myosin at the edge, understand the binding to the substrate, etc. So that's basically prediction of this active stress. And also you can actually measure the same for the active stress anisotropy. So now if I want to understand this, so basically so far I showed you this. That's the first plot, and now I want to show you this M alpha Q. Okay, so this is the anisotropic active stress. The magnitude is going to be dictated by m's times the nematic order parameter, which is hidden here, times alpha, but alpha is a constant, so it doesn't really matter. And then there is going to be an information about the orientation, right, which is also hidden in Q. Okay, so that's what you see here. So a little bit of orientation in the posterior, more myosin in the posterior, and then you let it go, and basically you see basically how the cables will reorient. There is going to be a lot of anisotropic active stress at the streak, and the cables are mostly aligned perpendicular to the streak. Okay, so this is basically m times s. This is the orientation of the cable. If you do the experiments, you see something very similar. So basically there is a lot of alignment of the cables perpendicular to the streak. Okay, so we're happy to compare basically velocities or models from from experiments and model, also the active stress. But what we wanted to do, we wanted to reproduce the attractors and the repellers. Okay, so now we said, okay, what I can do, I can just use this velocity field and advect the Lagrangian grid. So I start with a uniform grid, and then the later stage I get basically this distribution of the grid. Okay, so this is what I get from the velocity of the model, and you can do the same from the velocity of the experiment, and you get something similar. Okay, so that's basically the embryo has changed shape. Okay, and now you can compute the repellers. If I compute the repellers from the model, there are going to be two repellers. One that is very strong, is separating the anterior part and the posterior part of the streak. That's repeller number 2. And this is repeller number 1, that is separating extraembryonic with embryonic, and of course is more demarcated where the pair actually shrinks. And you can see it from here that there is a larger stretching of these rectangles compared to here. Okay, so there is still going to be a relative separation. You see basically that this guy is expanding, this guy is contracting, so there is going to be basically a separation, but this separation is higher in the posterior because the pair is basically shrinking. Compute the same for the experimental velocity and you get something similar. So you get repeller number 1 and repeller number 2. Okay? Okay, so now we want to understand how do they form. Can I actually deconstruct these repellers and understand what forces are, or what combination of forces are generating those, okay? And can we control them? Okay, so this is now gonna be the uncontrolled or the wild type, and now I'm gonna start deconstructing these repellers. So for repeller number 2, so we wanted to eliminate repeller number 2. And we thought that basically repeller number 2 intuitively is associated with the myosin, with the mesoderm that starts in the posterior, with the guys that do active intercalation. They will generate basically convergent extension and they will elongate the embryo. So what we did basically in the model, we just left everything unaltered, but we basically eliminated this myosin in the posterior, this myosin bump in the posterior. So remember that at the beginning, there was an additional myosin bump in the posterior. So what we did was just, let me eliminate this initial condition, which is eliminating the mesoderm, and solve the problem as is. And if you solve it, basically the model predicts that the embryo stays circular, and now the repeller number 1 becomes circularly symmetric, and actually separates the extraembryonic cells from the embryonic cells. Because I removed basically this myosin, I let the model run, and the model predicts that there is going to be a circularly symmetric repeller, and the embryo keeps a circular shape. Okay, same parameters, the only thing I just changed is this initial condition. Now the collaborators basically, they have an FGF receptor inhibitor. So if you basically add this FGF receptor inhibitor, you block mesoderm induction, so therefore you eliminate mesoderm cells. Okay, so you go to the embryo, do this drug, and you do the same experiments, and the embryo with this drug, no mesoderm, stays circular and develops a repeller that is circularly symmetric compared to the wild type. Okay, so basically this means basically the repeller number 2 is implicated with embryo shape change. Okay, because now the embryo is not changing shape. Okay, so that's basically what we were able to show. Now we say, okay, can we actually eliminate repeller number 1 and keep repeller number 2? For repeller number 1, remember, is this that separates embryonic with extraembryonic. We said, okay, this is actually caused by a competition between these cells that are pulling and stretching the embryo, the extraembryonic cells that are basically pulling away up, pulling out, and the myosin contractility inside the embryo. Because, isotropic myosin contracts, so you have a force that contracts, another one that pulls, their balance will generate this repeller. Okay? So what we did basically in the model, we said, okay, I'm gonna just turn off the boundary conditions of the velocity, and I leave everything unaltered. Okay? I do that, and then the model reproduces embryo shape change, but in this case, the size of the embryo decreased, because basically there is no pulling from these outermost cells. Okay, so this is the embryo basically retains shape change, develops repeller number 2, but eliminates repeller number 1. Okay, and our collaborator actually, this experiment was hard to do. What he was able to do to mimic this was basically burning or cauterizing the extraembryonic regions, or where actually the vitelline membrane, which is the regions where these cells attach, and therefore if you burn them, they cannot attach anymore, so this really imposes V equal to 0 experimentally. Yeah.
What exactly burned? I did not get.
They burned the vitelline membrane. So remember, I showed you that the outermost region of this system attaches to this substrate, and that's where cells start moving. Okay, so basically it burned this part, so these cells basically cannot go anywhere. It's just a wall. Okay, that's basically the black part here.
So if I understand, so the velocity has been set 0 when you said VB.
No, v equal to 0, just a boundary condition. We just replace that. The rest is the same of the model.
Okay, that's why it is, because if you treat with some drug, then it becomes tricky. Then it can affect the velocity.
Yeah, there is no way you can, it's also, people have tried to, this is a very challenging system because people have tried to basically cut part of the cells that are actually crawling. They are so smart that they redifferentiate into cells that crawl and then they keep pulling again. And this happens basically very asymmetrically around the embryo. Some cells will take less time to redifferentiate. So you're going to have very, Guillermo did basically a very smart experiment, I think, that he said he burned it and this was really imposing the velocity as in the model. Okay, so now this basically retains repair number 2 and eliminated repair number 1. Okay, but the embryo became smaller. Okay, so we basically thought the repeller number 1 is important to regulate, is a contributor to regulate embryo shape, size. Okay, and of course you can do basically the same. You can combine the two treatments, and you can have basically nothing. You have 0 repellers both in the model and in the experiment. So if you basically block epibole and also eliminate mesoderm, nothing happens basically in this system. So this basically was interesting because we sort of just guided by these coherent structures, we were able to understand that repeller 1 is arising from a tug of war between the embryo constriction due to myosin and the extraembryonic crawling. Okay, and this is important to regulate embryo size, so the size of the embryo. Why embryo size is important? There are several speculations. We have basically a comment in the supplementary section, but embryo size is very important if you have a patterning mechanism. If you have patterning equations or processes to say, okay, now I have to have adduction, reaction, diffusion, etc. If you mess up the size, you're gonna have very different Turing patterning or patterning situation.
Number of somites and all.
Yeah, everything, right? This is, so that's our speculation. Of course, it's very hard to test. It's a sort of an an evolutionary question. Say why the embryo would like to regulate its size.
These guys have done that. They cut the embryo, so effectively you reduce the size. You can cut the embryo and then you can look at the growth. Means it is not exactly same.
Yeah.
They see that the structures differ.
No, for sure. It's very likely that size is going to affect patterning. And repeller number 2 is instead arising from the active intercalation of the mesoderm and is associated with the embryo shape. That's the guy that transforms a disc into a pair. And this was basically, it looks maybe a little bit straightforward a posteriori, but it was really driven by these attractors and repellers because we were after them. Say, okay, it's cool. I understand how they form. Can we actually control them? And then we discovered this modularity. Because, for instance, it wasn't clear, at least from my understanding on the literature, that when you block mesoderm, people think in this community that everything stops. But instead, the active contractility of myosin, the isotropic myosin in the embryo, stayed to regulate the embryo size. That was a very surprising biological new result. And the fact also that you can knock mesoderm and the embryo still retains its size is a cool thing. So basically we're able to understand how these repairs form and also control them independently. So, yeah.
So for the second case where you remove the repeller one, so basically you're setting that at those points the embryo is not going outside the overall.
Yeah.
So how does that affect the size basically just to get?
Because think about it basically. Think about a disk of cells. These cells are contracting, but you're going to experience a pulling force. The pulling force would like to stretch the embryo, so it would like to actually increase the area.
Okay. So you're trying to contract, but someone is pulling, so you overall grow.
Yeah, but Yeah.
when stretching is not there, so you will just shrink and then
Yeah, exactly. Yeah. That's the thing. Of course, it's more complicated than that
Yeah,
because
but
basically,
just-
yeah. Okay, so this is basically control of repellers. And a few years ago, actually, this was a work that we did also with Maha when I was still, in Boston. We were also able to control the attractor in a very exotic way. So basically, this is the attractor, right? The streak. That's the model. And then we were able to control the attractor in many different ways. This is basically spontaneous twin development. So the attractor now is made by two streaks that try to fuse. You can get it in the experiment and in the model. We were able actually to control this attractor in the cheek from a line to a ring. Okay, so basically, let me show you. So this is basically the model and the experiment. Again, what we did here is the same stuff that we did basically here. We played with the initial conditions and some parameters that are biologically interpretable, and you can implement them in the lab. And with the same mechanical model or your mechanochemical model, you can actually reproduce very exotic perturbations. So to give you a more visual interpretation, the cheek develops this deformation pattern, but we were able to manipulate the cheek in a way that develops this deformation pattern, in which basically now instead of having an anterior-posterior axis, a streak, you're going to have a ring, and the ingression is on the ring, okay? Or even into a point attractor, okay? So, you can also transform this and have that everything contracts to the center, both in the model and experiment. Okay, so this is basically controlling attractors and repellers. I'll do a mini summary and then I'll add 2 more slides. So what did we actually learn is that these coherent structures allow you to compress spatiotemporal velocity into discrete units, attractors and repellers. Then we were able to build an active matter model that can explain these flows, the active stress patterns, and this dynamic embryo geometry, size and shape in a modular way. And then we learned that repellers, they control basically embryo size. Repeller 1, the repeller 2 controls embryo shape. And this is basically an interesting way to discover modularity in these systems. So this is basically KISS, is the guy who basically taught me the little biology I know, but all the credit goes to KISS. And also Guillermo was a student with KISS at the beginning and now is a postdoc in Cambridge, and Alex is a student in the group. So basically this to say that attractors and repellers, they are not only a way to compress and classify flows, but they can also guide you to build models, okay, and design perturbations, etc. I want to also, this is basically advertise a couple of more things. So if you want to compute attractors and repellers now on curved surfaces, dynamic surfaces, so this is basically beating zebrafish heart, experimental images. This is a pancreatic spheroid. This is basically active nematics on exotic, curved surfaces. You can now compute all these structures when your flows on, these different geometries. And we also have, the codes available. They are basically, I think we share it with you. I think that's it. That's all I have for the morning. Yeah.
The last from the curve surface.
Yeah. Yeah.
So this is on what surface? It's what system that represents?
This is to show basically that, this is to show that they can compute. So this is the simulations were from the group of David Santillan at UC San Diego, and they now can solve active nematic flows on curves on any geometry. Yeah. So now you can, the interesting thing, I don't know where did it go, but the interesting thing that now you can start looking at, it's fine, I don't need the slides, but in general, you can now start understanding, so there are a couple of interesting things. First of all, in organogenesis, you have flows on curved surfaces that are themselves evolving, so how do you do this analysis? And the other thing is that now, when you have curvature and curved surfaces, you can also start understanding what's the effect of the curvature of the surface on, I don't know, the flows, the strain, and stuff like that. And you can also use this for doing control. So, because on particular curved surfaces, the motion of topological defects are of a particular type. This will steer the motion of material transport, etc. So this is now a way to attack basically these problems. Yeah. And one more thing, guys, in the talk that is gonna be during the conference, I will talk about how these attractors and repellers are also important for understanding advection reaction diffusion. So if you have basically advection reaction diffusion on top of flows, which is very typical, for instance, in development, but in many systems that are deforming, you will see basically that very elegantly these attractors and repellers and Cauchy green will modulate diffusive fluxes. Okay, so this is something I think I'll discuss on Monday or Tuesday. Yeah. Okay.
Thank you.
Okay.
Questions?
That's good, yeah. If there are no questions, it was clear, or you're shy.
I think because for many of them, maybe they're seeing this kind of system for the first time. So
Yeah. But I mean, basically, it's a beautiful connection with the equations that Novan was saying, basically, of the similar family. Yeah.
But important things one should notice that, the experiments are how beautifully done, but how to interpret them, right?
Yeah.
How to dissect each part. I mean, that's how beautifully it was done. Okay, what is responsible for this part, like one kind of repeller, what is responsible for other kind of repeller. Also designing it. I mean, this takes a lot of thinking and a lot of time. And of course, they managed to figure it out.
Yeah, it took, I mean, this basically, we've been working on this system for a few years now. Yeah. All right, that's
Okay,
good.
Let's take a break. We'll go for tea, coffee, and then we'll come back at 11:30.
Yeah.
Backward in time also the system will retain its original.
Well, which system? Tell me.
The attractors and repellers that we have seen.
Yeah, so we were discussing this yesterday. So if you, yes, because basically these are just accounting for motion. Okay. Now the way to think about it, maybe after the discussion of yesterday, is that if you give a velocity field and you pick the initial condition, someone gives you a velocity field. Let's say the measured velocity field of the embryo. Okay, of that embryo. Okay, and you advect an initial position later in time, and then you advect it back in the same time interval. You go to the same initial position. Okay, so that's basically because you have a velocity field that is deterministic. Now, the reversibility that creates confusion is that eventually you are thinking about something that advects and diffuse, like a parcel that advects and also diffuse. Okay.
The alignment also written exactly back to its original state.
Yeah, the alignment I don't know. So you're talking about is this equation which equation? So the PDEs are not reversible, but the PDEs are describing the evolution of the velocity or the Q tensor.
Velocities we are getting that from PIV, right? The data that we have.
Yeah, so you're talking about the model or the experiments?
The model.
Okay, the model. The model also predicts the velocity because there was the model and the experiment, right?
But we have three equations, right? One is with the governing M, and we have one with the governing Q.
Yeah.
And one is the force balance.
Yeah, that gives you B.
That gives us B.
Yeah.
But S is also unknown, right?
S is the nematic. Yeah, SS is in Q.
But we have only three conditions,
right? No. Q has 2 degrees of freedom, basically S and the angle. So we have 4 equations for unknowns. Yeah, we need to have 4 equations for unknowns. Yeah. I mean, 2 velocity, 1 m, and Q that has S and the angle. So we are not input.
Q and S are kind of interdependent.
Yeah, they are interdependent. Yeah.
You get it from the director field. Like you define N, and if you can get a. So of course, the vector sum could be 0. That means there is no ordering, but still magnitude is there. So that magnitude gives you the isotropic part, and the vector sum, if it is finite, then that is going to give you the ordered structure, the anisotropic part, and then you break them in two parts. So, and you have, yeah, that's how it comes. And then you have to maintain, like, Q should be, anisotropic part should be, the trace of anisotropic part should be 0. And you know how should be an
that's all built in.
So once you take care of those things, you will start getting the magnitudes for the isotropic and anisotropic part. So if we know the structure how it should be, we have the director field, like the coarse grain structure, that the yellow color vectors were there, right? So that's your director field.
Yeah, from the experiments. Yeah.
And of course you can do the same
for the model. Yeah.
So yeah, that's how it is. But yeah.
Okay.
I'm not learned with pneumatic fields.
No, it's a cute system. Yeah.